So for me, it's a great honor to receive today Dr. Ana Clara Bobadilla. She is from the University of Wyoming, from the School of Pharmacy, Health Science, from the Pharmaceutical Science Division Department. I want to tell you a little bit about her career. Her education is her BS from awards with honors from the University of Pierre and Marie Curie. Also a master's degree in neuroscience major, also from the University of Pierre and Marie Curie, and her PhD in neuroscience with honors, also at the Pierre Marie Curie University, now the Sorbonne University. She had research appointments as an assistant professor in the School of Pharmacy at the University of Wyoming, also a research assistant professor at the Department of Neuroscience in Medical University of South Carolina in 2019. In 2014, she obtained a postdoctoral research fellow at the Department of Neuroscience at also the Medical University of South Carolina. She had several publications all over the years, conferences, also mentorship, participation in fundings and awards, several travel awards from different societies and academies. Also, she has some teaching experience also in psychopharmacology, techniques in neuroscience. Also, she's been directing or advising undergraduate students also, reviewerships in several journals, additional training as a digital teaching and learning course, also some career development programs. She had also leadership, science, advocacy, and community involvement. And I want to mention this because I think it's very, very important. As an associate of the Professional Women's Nexus, an organization collecting and disseminating gender-related information to a broad network of scientists. Also as a volunteer at the Girls' Days of Summer Camp, and volunteer at the Center for Women, a non-profit organization considering women in the Low Country area, and also several media. I can tell you that she has a fantastic career. I admire her a lot. So I thank her, Ana Clara, for being here today, for accepting this invitation. And thank you very much. The stage is all yours.
Thank you. Thank you for that really nice introduction. I appreciate it. So let me share my screen. There you go. Can you all see the PowerPoint presentation?
Yes, we can see it.
Awesome. So I would like to start by thanking Dr. Alfonso Diaz-Furlong for inviting me here today to share my work, and also everybody who joined in. Thanks for your time. I really appreciate it. Today, I'm going to talk about how the brain processes the seeking of natural and drug rewards in a very specific region within the reward circuitry in the brain called the Nucleus accumbens core. And so this is one of the questions that my lab is interested in. The lab is very, very young and new. We are a very small team. We moved to Wyoming a couple of months back in the middle of the COVID pandemic, so the perfect time to start a lab. Right now we have the lab manager, Lucio, and then a couple of undergrad students, Natalie and Cameron, and the graduate student, Carl, just started his rotation in our lab, so we're very excited to have him. And we are hiring at all levels. So if you are looking for a postdoc, for a postbac, if you want to start a PhD program, or you're looking for a tech position, please, and any of the things that I'm going to talk about is in interest for you, just please reach out and I'll be happy to talk more. So the lab, what do we do? We have two main scientific questions. The first one is the one I'm gonna talk about today, is how does the brain choose between different types of reward? And what I mean by that is I'm gonna compare how the brain seeks cocaine, a drug of abuse, and natural reward, so sugar or sucrose. But the other question that we're also interested in lab is to see if natural rewards can become addictive. And so what I mean by that is that you probably have someone in your family or your friends or even yourself who have said in the past, oh, I'm addicted to chocolate, or, you know, I'm addicted to the gym or something like that. And it's true that if we take the example of sugar, for example, there are a lot of similarities sometimes with what the behavior we see in addiction related to drugs of abuse. We crave sugar. We often eat more than what we intended to at the beginning. We also keep eating despite negative consequences, known negative consequences. And you can see here in the picture of this little girl on cotton candy, this sugar rush is very similar to what we can feel when we have stimulants like cocaine or amphetamine, you know, this very high energy state. So we're trying to see if at the cellular and molecular level, the certain natural rewards in certain contexts can have the similar molecular mechanism that drugs of abuse have. And so to start us off, I would like to spend a little bit of time on the idea of reward-based positive reinforcement. So this is an evolutionary survival strategy that has been very successful and is shared by many species. And so the idea is that natural rewards, so the things that keep the species alive, like, you know, eating, drinking, reproducing, or nurturing, are going to induce pleasurable feelings. And those pleasurable feelings are in turn going to reinforce the behavior and going to induce a repetition of the behavior. But in certain cases, like in substance use or in eating disorders, a specific drug or a very specific pattern of eating is going to induce both pleasurable and unpleasant feelings depending on the drug, depending on the stage of the disorder. And those are also going to reinforce the behavior, but at the expense of the natural rewards and the behaviors that keep us alive. So it can become problematic. And in the addiction field, you often hear or you see when you read the introduction of addiction papers, the authors often say the drugs of abuse hijack the natural reward circuitry. And although we know that a lot of different regions in the brain are overlapping between different types of reward, and also the release of certain neurotransmitters, so molecules in the brain are also the same when the brain answers to different rewards. We really wanted to answer this question of, do drugs really, drugs of abuse, actually hijack the natural reward circuitry at a network level? So is the same networks of cells that respond to cocaine are going to be the same ones that respond to sucrose? And so to answer that question, we use a very widely used model that is called the reward self-administration and relapse model. So here, the example that I'm going to give you is with cocaine. So you have this mouse in this operand box. It has two nose pokes, so it's like basically holes where they can just poke their little nose. You have an inactive nose poke and an active nose poke. And so every time the mouse pokes on the active nose poke, it's going to receive an IV injection of cocaine, so directly in the veins through a catheter that we implanted in the jugular vein. And so it's going to do that, and at the same time that it gets the cocaine infusion, he's going to be exposed to cues. So we have a light cue inside of the nose poke, and we also have a sound cue that comes out of a speaker. And so the animals, the mice that are very smart, are going to realize very quickly that it's much better to poke on the active nose poke, on the active port here in black, compared to the inactive one. When you poke on the inactive one, nothing happens. So the animal understands very quickly that if it pokes on the active nose poke, it's going to get the cocaine paired with the cue. And so we're going to have this phase of the model that is called acquisition of self-administration. And around day 10, 12, the animal is going to stabilize the infusion. So you're going to get, like a very stable number of infusions at each session. And so the fact of pairing this reward delivery with the cue is going to associate the two in a way that the animal is gonna, every time he sees the cue, he's gonna think of the reward, and every time he gets the reward, he's gonna see the cue. And so once we are done with this acquisition phase, we move to what we call the extinction phase. So here the rules are changed. None of the nose pokes are actually active. So even if the animal pokes on the active nose poke, he's not going to get the cocaine or the cue anymore. Nothing happens. So the animal rapidly learns that, well, there is no point in keep poking on the active if nothing happens, so I'm just going to stop. So that's what we call extinguishing the behavior, because the animal is going to stop interacting with the nose pokes. And then what we want to see is the seeking. We want to look at the brain during seeking. So how do we do that? How do we induce seeking? Well, we could give cocaine. That would induce seeking. But the problem with that is that if you do that, you're going to induce a lot of release of neurotransmitters in the brain, and it's going to become really messy and complicated for us to study. So what we're going to do, instead of giving the cocaine, is just show the cue that was associated to the cocaine during self-administration. And that, what we think happens is the animal thinks, oh, the cue is back, the cocaine is back, let me go and poke on the active nose poke. And that's what they do. On the reinstatement day, it's called the relapse model, it's called the reinstatement day test. And so we have here a very robust increase of nose poking in what was the active port. And so this kind of is how we model the brain, and we induce the seeking. We model the relapse and we induce the seeking. And usually the way that you see reinstatement day presented is by looking at the number of active nose pokes. You compare during the last days of extinction around here, so it's very low, to the reinstatement day, and you see that you have a really robust response. So this is the seeking of the reward. And so using this model, but also many models where the drug is given in a more passive way, we call that non-contingent, or in a contingent way like self-administration, the field has identified the reward pathway inside of the brain. So this is a very simplified circuitry. It's way more complex than this. But here you can already see some of the main players, the main regions that are inside of this reward pathway. We have the prefrontal cortex, the hippocampus, the nucleus accumbens core, and the nucleus accumbens in general. That's the region we're going to focus on today. The amygdala and the ventral tegmental area. So all of these regions are included in the reward pathway. And even in this very simplified representation of the pathway, if you start looking at how these regions communicate between each other, it can become very complex really quickly. You have dopamine release from the ventral tegmental area to all of the other regions of the reward pathway. And so this dopamine is very important for reinforcement, reward, and also emotional arousal. You have also a feedback, a negative feedback loop that goes from the nucleus accumbens to the ventral tegmental area, and it releases GABA, so that's why it's a negative feedback. And then, on top of all of that, you also have glutamatergic transmission that goes from almost all of the regions of the brain, of the reward pathway, into each other. And so we focus on the nucleus accumbens. And so why we do that? Well, because it's a region that has a lot of afferents, so receives, and efferents, so projects different connections, and is really at a key central place within the reward pathway to be able to treat information about reward. And particularly, a lot of studies have shown that the glutamatergic transmission from the prefrontal cortex to the nucleus accumbens is very essential, is really essential for seeking. If you disrupt this glutamatergic transmission, if you inhibit the PFC, or you cut the transmission, or you block glutamate at the level of the nucleus accumbens, you're never going to see seeking, no matter what you do. So we know that this nucleus accumbens is very important for seeking. And so we approach the nucleus accumbens instead of looking at it as a very homogeneous region with all of the same cells. We try to look at it as a network of cell populations. So what do I mean by that? If we focus now on the nucleus accumbens, a long time ago, Simon introduced this idea of the engram cells. And what he meant by that is that a very specific number, a very small number of neurons that are activated at the same time and that carry the memory trace. That's how he defined the engrams. And a little bit later, Donald Hebb developed this idea into the idea of ensembles. And he said, okay, when we have a memory or when we are doing a task or when we are just learning something new or doing something that we already learned, we have these very small amount of cells, so this network of cells, ensemble of neurons, that not only is activated at the same time, but is also projects into each other. You know, you have like this coordinated firing that is also projecting into each other. So that's where the idea of neurons that wire together, fire together come from. And since then, a lot of different labs, like the ones led by Regina Carelli or Bruce Hope and Yavin Shaham at the National Institute on Drug Abuse, NIDA, in the United States, have really done fantastic work showing that you can have drug-linked ensembles in different places of the brain. And they came up with this idea, this concept of mechanistic resolution. And it kind of proposed to explain the brain's ability to encode and distinguish between very different stimuli and cues. So let's take an example to try to understand better how it works. Here you have a random region inside of the brain that has 300 neurons, okay? If we determine that our ensemble in this region is 1% of the neurons, which again, like a very, very small percentage of neurons compared to the rest of the area, how many possibilities of different ensembles you're going to be able to have? So this is a question that I would like to ask you. If you go to the poll everywhere that is in the chat, so I think you can see it in the chat probably. There you go. Thank you, Alfonso, for putting it again. So if you could please go there, and I'm going to activate the poll. There you go. And so we should be able to see. So take your time and try to do the math. And I know it's not easy, like I'm asking you to do it, but I know it's not something easy at all. So let me know if you're not able to see the poll. I think that it's activated. So I don't record any response right now. So right now we are at 0. I don't know if you guys are able to the share window. Okay.
Ana Clara,
Yeah.
I have the poll open, but for me, I don't know if for everybody, but I cannot respond.
What about now?
I don't know if anybody in the chat can tell us if you can respond to the poll.
Let me see.
Yes, they say that this poll is locked.
Okay, it's locked. So I'm gonna unlock and then maybe now it should work.
Yeah, yes, it
Oh,
works. Okay. Yes, you can answer now.
Right. Yeah, thank you. Thank you for letting me know for the lock thing.
No, thank you very much, Ana Clara.
Okay, so we're gonna turn this on. Okay, and we see if we go to so right now I'm seeing your responses. I don't know if you guys can see them. So I'm having a lot of C's right now, a few A's, a few B's. So we have, just to give an order like of amplitude of how many possible combinations of different ensembles we can have. So you have like around 4,400, 3,000, or over 4 million of possible combinations. Okay, so it's going to give you a few more minutes. Yeah, I think that we have. So I tried to put this on the results, but it doesn't seem to show. So I'm just going to read it to you. Basically, we have 63% of people who answered who said that we have over 4 million possible combinations. And you guys are really smart because that's exactly the response. I don't know if you can see back to the PowerPoint presentation. Are we back to it? Is that okay? Can everybody?
Yes.
Okay. So this was the math that you were supposed to do. So don't ask me to try to explain this math. I would be completely unable to. But just to give you an idea of how much we're able to learn and remember, we will have more than 4 million possible combinations if we only take 1% of the neurons. And remember that this is just one region of 300 neurons. You have in the brain many regions that are, carry way more than 300 neurons. So this is just to give you an idea of how limitless our brain is when regarding the ability to remember things and learn new things. So now that we have defined our ensembles, how do we see them in the brain of animals? So we use genetically modified mice. So the ones we use are called CFOS trap mice. The trap stands for targeted recombination in active populations. And I'm going to explain you how it works. So we have this CFOS trap mice. CFOS is an immediate early gene. And so what it means is that every time the cell, so the neuron in our case, is activated, you're going to have expression of CFOS right away. So that's kind of a way to see if the cell is activated or not. So we have the CFOS promoter, and following the promoter, you have a modified CRE. So this CRE is linked to the estrogen, a modified estrogen receptor that is sensitive to tamoxifen. Tamoxifen is just a molecule that we're going to use to open our window and tag our ensembles. And so we also breeded this CFOS trap with another type of genetically modified mice called the IE14 reporter mice. And so these mice carry an allele designed to include this LOXP, flanked stop codon. So what does it mean? It means that you have everywhere in all the cells a stop codon, so it's not gonna be read and expressed, and then a TD tomato. And so what happens when the cell is activated but there is no tamoxifen? You have expression of CFOS and you have the expression of the modified CRE. But because there is no tamoxifen, the CRE remains at the membrane with the modified estrogen receptor that is at the membrane as well. And so the CRE cannot come and cut here the stop codon. This is a CRE-LOX model. And so usually the CRE recognizes these LOXP sites and is able to cut. But the CRE is stuck outside of the nucleus with the estrogen receptor. So there is no tomato expression. The TD tomato is just a fluorescent marker that we can see when we look under the microscope. But what happens if the cell is activated and we put the tamoxifen in there? So we open our window. Then we're going to have expression of CFOS, expression of the modified CRE linked to the estrogen receptor. But this time, the tamoxifen is going to come linked to the estrogen receptor. The CRE that was locked outside is going to translocate to the nucleus. It's going to recognize the LOXP sites, cut them, and remove the stop. And so that means that the cells that were activated when the tamoxifen is present, so when we open our window, is going to express the fluorescent marker TD tomato forever, permanently. So if you have to remember anything from this slide or from this mice, is that every time we put tamoxifen, we tag all of the cells that are expressing CFOS in red permanently. And so that's how we see our ensembles. And so using this technique, we're going to be able to tag within the same animal the ensembles that are activated when the animal seeks cocaine versus when the animal seeks sucrose. But before doing that, we needed to be sure that this model, these CFOS trap mice, were able to be used in self-administration, and that we're able to actually see something, see the ensembles in our region of interest, which is the Nucleus Accumbens core. So we started with a very simple, very standard single cocaine self-administration, exactly the same one that I introduced to you a little bit early on. 10 days of single cocaine self-administration, extinction training, where the animal extinguished the behavior. And then we have two very rapid and short induced reinstatements. In the first one, we're going to show the cue. So the animal, remember, is going to start seeking and it's going to start nose poking on the active port again. And that's where we're going to put our tamoxifen. So that's when we open our window. So all of the cells that are activated during this cue induced reinstatement are going to be tagged in red. And then we do a few more days of extinction. This allows two things. First, we allow the behavior of the animal to go back down to an extinguished level. And that's important. If we want to see the increase again, we need to go down first. And then we also going to allow for the cell to produce the fluorescent marker TD tomato. It needs a few days to be expressed and visible. And so we have a second cue-induced reinstatement. And this time we're just going to do an immunohistochemistry. So we're going to tag the CFOS protein as well as NEWN protein, just to tag the neurons that we see, right? So what does that mean in behavior? In behavior, it means that we have the cocaine self-administration. We have males and female mice. They both self-administer really well, and they're able to discriminate between the active and the inactive ports. Then we do the extinction training. We have really here a very nice extinction burst. So this is the first day of extinction when they're not getting the cocaine anymore. They're like, hey, what's going on? Like, there's no more cocaine. So they start like nose poking like crazy on the active one because they don't understand why there is no more cocaine coming. But then after the first day, they extinguish the behavior. There is an equal cocaine intake between sexes. You know that the National Institute of Health is really, in the United States, is really pushing for investigators to look at both males and females, because a lot of studies were done in males and females were not accounted for. And so all the treatments worked very differently in women and men. And so it's very important to try to introduce not only women in science, but also female mice in our experiments. And so we have equal cocaine intake between the sexes. After the first reinstatement, when we put the tamoxifen, we also see that the seeking is similar between females and males. Okay, so we have this reinstatement day compared to the extinction that we saw earlier. That's very nice. Since we do repeated reinstatements, we want to make sure that the seeking level at this first reinstatement is the same one at the second, because remember that here during the cue-induced reinstatement, we show the cue, but we don't give the cocaine. So at some point, after the initial, rush for answering to the active no-spoke, the animal is feeling that the cocaine is not there. So what could happen is that you have what we call a within session extinction, meaning the cue loses its balance, its positive balance. And so we try to avoid that. That's why we did very short reinstatements, right? And we made sure that for both males and females, they responded at the same level during the first reinstatement and the second reinstatement. So this is what we call a no ordering effect between the reinstatement. The fact that the first one is the same as the second one. And so once we do the 2 reinstatements, we sacrifice the animal at the end of the experiment, and we do the immunohistochemistry for the CFOS protein and the NEU-N neuronal marker. What we see is this. We focus in this region that I told you about, the nucleus accumbens. It's really nice because you have the anterior commissure here that really helps you localize the core. And then you have some tomato. So it's called tomato because it's in red, the fluorescence. So you have the tomato cells. So these are the cells that were activated during the first reinstatement. You have the CFOS positive cells. So these are the cells that are activated during the second reinstatement. And then you have all of the neurons of the area that are marked with a NEU-N. And so you see that some cells are both tomato and CFOS. That gives the yellow. Some cells are only tomato, and some cells are only CFOS. So we're going to try to quantify all that and see how much we have. So that's what we did. First, we need to start with a few controls just to make sure that we're actually tagging what we want to tag. And so we had this vehicle group where instead of giving tamoxifen and opening our window, we just gave vehicle. And we were reassured when we saw that we saw almost no trapped cells or no tomato positive cells in this group. Then we gave tamoxifen to open the window and tag the ensembles, but instead of inducing a cue-induced reinstatement, we left the animal in the home cage. And when you leave the animal in the home cage, it's a very familiar environment. So you don't expect a lot of cells to be activated. And that's what we see here. So we see some trap cells, so cells that were activated at that moment, but it's a very low percentage, almost, not significantly different from 0. And then we did another group where instead of doing the cue reinstatement, we just opened the window during one of the last extinction sessions. And then we see this extinction ensemble. So the cells are activated when the animal is actually extinguishing, so not seeking the drug. And so here we see a significant amount of cells that were trapped, that expressed tomato. This is our extinction ensemble. And this is the interesting group. Finally, the last one that we do. And we see that when we tag cells with tamoxifen after the first cue-induced reinstatement, we also see around 1%, so 0.7% of cells that are activated. So this is our cocaine-seeking ensemble, okay? And we were very interested in these two because we know that different types of cells are believed to, drive extinction or drive reinstatement. Some of you may know that in the region of interest that we're looking at, the nucleus accumbens core, it's mostly composed by what we call medium spiny neurons. So these are GABAergic projection neurons, and they can express a different type of receptors to dopamine. The type 1 dopamine receptor or type 2 dopamine receptor. And depending on if you are a D1 MSN or a D2 MSN, your projections go to different places, what we call the direct and indirect pathway. So this is very true in the dorsal part of the nucleus of the striatum. Not so much in the nucleus accumbens core and in the ventral part. Like many papers have shown that you don't only have D1s that go into direct pathway, but the D1s can also go into the indirect pathway. But overall, you see that when you activate the direct pathway, you're going to have kind of a go signal. And when you activate the indirect pathway, you're going to have a no-go, so like a stop, right? And so the D1s are going to be involved in increasing of seeking, and the D2s are going to decrease seeking or even induce, how do you say, when you, aversion, it's going to induce aversion. And so knowing this, that the D1s are more associated with seeking and the D2 can be aversion, then my next question that I'm going to try to unlock right now is, what do you think, what type of MSNs are recruited in each ensemble? So I'm going to unlock and activate. So you can go to the same website that was in the chat that Alfonso wrote earlier, and it should be activated for you to answer the question. Activate. There you go, and then it should be unlocked. So let me know if you're not able to answer.
It is activated. Thank you.
Good. Thank you. So you have kind of like, you have to go through the answers, right? So the question is, what type of MSN, either D1 or D2, are recruited in the seeking or extinction ensemble? So you can be mostly D1s for both type of ensembles. It can be mostly D2s for both type of ensembles. It can be 50-50. You have the same amount of D1 or D2s. And then you can have, for the seeking ensemble, mostly D1s. And for the extinction ensemble, mostly D2s, or vice versa. D2s for the seeking ensemble and D1s for the extinction ensemble. So I'm starting to see some responses, so that's good. So this one takes a little bit of time to realize. And just as you respond, I would like to just address this picture here. So these are genetically modified mice, different than the ones that we're working on today, that express the protein GFP, the fluorescent protein GFP in the D2 MSNs, or the tomato again on the D1. And so you see that when you merge these two, you usually don't overlap. You know, you have a very, very small amount of medium spiny neurons that express both D1 and D2, but it's really minimal. If you're an MSN, a medium spiny neuron, you express either D1 or D2. That kind of like characterizes your life. Okay, so I'm seeing some responses. That's good. I'm going to give a few more seconds. Right now, I have a majority of people, 44%, that answered D, that you're going to have some D1 MSNs in the seeking ensemble and some D2s in the extinction ensemble. Now it's up to 50%. And there you go. It's not moving anymore. And so, yeah, so basically you guys got it. It's over 50% of people responded D. And so we're back to our PowerPoint. We have the seeking ensemble are mostly D1s and the extinction ensemble are mostly D2s. And how do we know that? Well, we did this experiment called the RNA-scope that is going to allow us to tag all the tomato cells and also tag the cells that express D1 or D2. So here, to make it a little bit more complicated than it already is, the tomatoes are in green instead of in red. The D1 MSNs are in red and the D2 MSNs are in blue. And so you see that some tomatoes are going to be overlap with the D2. So those are going to be D2 tomatoes. And then some tomatoes are going to overlap with the D1, and these are going to be D1 MSNs. And so what we see is that in the seeking ensemble, you have the majority of cells that are identified as D1, whether in the extinction ensemble, you're going to have a majority of D2s. So this really fits the literature really well, where you have a majority of D1s in the seeking ensemble and a majority of D2s in the extinction. And so we did this But then remember, there is a second reinstatement, right? And so when we also needed to count the number of CFOS positive cells. And if you think about it, this tagging with tamoxifen depends on CFOS because the CFOS is the promoter that is in front of the CRE, remember? So what we expect is that all of the cells that are activated during the first reinstatement, and so that are tagged in red, are also going to be activated in the second reinstatement because it's the same behavior. So if we're talking about ensembles that are specific to the behavior, this should be activated as well. So what we see is that all of the groups, the CFOS is the same, around 4% of the cells express CFOS. And this makes sense because remember that the first test, the one we looked at here, is different for all the groups, but the second test is the same one, is the reinstatement. So we do have, for the same behavior, the same amount of cells that are activated. So that's good. But the interesting question is how many of these tomato cells are also CFOS cells? And so that's what we looked at. We looked at the percentage of overlap between the tomato positive cells and the CFOS positive cells. And we saw that only in the group that underwent 2 reinstatements, we have a significant overlap of around 50%, meaning 50% of the cells that are activated here are also activated here. And that's our seeking ensemble. And interestingly, we were able to correlate the overlap with the behavior. And so the higher the overlap, the more the tomato cells overlap with the CFOS positive cells, the stronger the reinstatement behavior, the more the animals are going to poke on the active lever. So this gives us a positive correlation between our measurements and the behavior. So it seems to be working. We did this with the cocaine single self-administration. We also did it with sucrose single self-administration, very similar results. I'm not going to show it for the interest of time, but it's very similar. We're able to identify a sucrose seeking ensemble. So now we move on to the model that is the more interesting for us, is to try to compare cocaine versus sucrose. So we had to modify a little bit the self-administration model because we needed to expose the animals to both rewards, right? So we have one day on the cocaine session, the animal is going to do single cocaine self-administration. And then the day after that, they're going to do sucrose self-administration. So it's the same idea. You're going to have an active nose poke, an inactive one, and this time instead of getting the cocaine IV, you're going to get a pellet of sucrose. So it's going to be ingested. And very important, there is one reward per day. So in the cocaine days, it's only cocaine. In the sucrose day, it's only sucrose. And we switch the active and the inactive nose poke in between rewards, meaning the active nose poke for the cocaine becomes inactive on a sucrose day, and the active one for the sucrose become inactive on the cocaine day. And so why do we do that? Because we wanted to make sure that the animal understands that these are different rewards. We just don't want him to go always to the same active nose poke, and the fact of nose poking becomes just something that is habitual and not really something that is like a goal-directed behavior. And also, if we switch the nose pokes, we're able to know what the animal is seeking, right? If it goes for the sucrose associated nose poke, we know it's seeking sucrose. If it goes for the cocaine, we know it's seeking cocaine. So we do that. We have an extinction training. And then we have different types of reinstatements you can do. You can just show the cue associated to the cocaine or the sucrose. And then you can show for the second reinstatement the other cue, the one associated with the sucrose or the cocaine. And then at the end, we also did kind of like a simultaneous cues test where we show the animal all of the cues, both cocaine and sucrose, and we let the animal decide what it seeks more. So what does it look at in the behavior? Well, at first, the animals have no idea what they're doing. They're kind of like nose poking a little bit everywhere, all over the place. But then pretty quickly, you see that they're able to discriminate between the active and the inactive for each session. So they know that for a cocaine session, so here's like the gray part, you have more pressing or the active. And then for a sucrose session, we also have discrimination between the active and the inactive. But what is really interesting is that you see this sawtooth shape here for the inactive, and every time it goes up for a sucrose session. So that means that even though the animals understand that this is a sucrose session and they're gonna they're gonna nose-poke more for sucrose, they are still nose-poking for the cocaine, kind of being like, oh, maybe, you know, I'm lucky and I can get a little bit of cocaine right in there. So for us, it kind of shows the greater motivational value of the cocaine compared to the sucrose. Then we looked at the infusion or the pellets, and it's more or less stable. We also looked at the intake. One of the things that we were worried about is that cocaine is an IV reward, whereas sucrose is ingested. So we thought, oh, maybe like these are very different ways of experiencing a reward. So maybe we're going to have exposure to one reward more than the other, and that can bias our results. But thankfully, when you put the males and the females together, they have the same amount of sucrose and cocaine. They take the same amount of sucrose and cocaine. Then the extinction session, we see that they extinguish really well. There's not one nose-poke that has a harder time being extinguished or anything like that. So then we go to the reinstatement. And when we show the cocaine cue, we see that the animal is going to nose-poke more on the cocaine-associated port. So that's great. Like we show them the cocaine cue, they seek cocaine. And when we show them the sucrose cue, they seek sucrose. It's significant, but you can see that the amplitude of the effect is quite different. It's much stronger for the cocaine than it is for sucrose, and that's something that I've always seen with mice. They don't reinstate for sucrose cues very well. So what people usually do is to give a few pellets of sucrose at the beginning of the test. But we didn't want to do this because we know that when we give the reward, that induces a lot of changes in the brain. So we just increased the N until we got enough to see something that was significant. So our cues are able to induce the type of seeking that we want. So now I have a question for you. What happens when we expose the animal to all of the cues, to the cocaine one or to the sucrose one? What reward do you think the animal is going to prefer? And so I invite you to go back to the poll everywhere website. I'm going to activate. I think it's activated. There you go. I'm going to show the responses, and you should be able to choose if you think that when we show all of the cues, the animal is going to go more for the cocaine cue or for the sucrose cue, or they're going to press for the same for both rewards equally. So I'm starting to see some responses. I'll let you think about it. What would you choose, the cocaine or the chocolate cake? Okay, interesting. We had there for a moment, same amount of people for all. It's interesting. Okay, so right now, a lot of people are saying, like the majority of people, 44% of people say that animals are going to go for sucrose when they are exposed to both cues. Okay, both records equally is catching up. Okay, a few more answers and we'll go back to the results, see who was right. This is like looking at the results in a political poll day, you know, election day. Okay, so we have more or less stable responses. 23% of you said that the animal was going to prefer cocaine cue. 38% said that they're going to prefer sucrose, and 38% both rewards equally. So let's see what we have. So we're back to the presentation. So when we show both cues simultaneously, so we are at this level, we see that the animals actually prefer the cocaine cue. And this is kind of surprising because a lot of studies have shown, work of Serge Ahmed and many people after him, that if you leave an animal, if you give the animal a choice between cocaine or sucrose and the reward is present, the big majority, the large majority of animals are going to go for the sucrose. So they're going to choose sucrose over cocaine most of the time. Sucrose over heroin, sucrose over almost any type of drug of abuse. But that is only when the reward is there. When you only show the cues associated with the reward, then the cocaine becomes the stronger cue. And so that kind of tells us that there is something that the cocaine does, an association with the cue, that becomes stronger than any other reward, or at least that the sucrose reward. And so that, like, if we understand how the cocaine does that, we kind of solve addiction, because, well, that means that we understand why cocaine and drugs of abuse have such a pull, have such a control on the seeking, right? And so we went back to tagging our ensembles. And so we did tamoxifen administration right after the cocaine cue. And then we measured the CFOS positive cells either after the same cocaine cue. So we should have a pretty big overlap because it's the same reinstatement for cocaine, or the sucrose seeking. And so we'll see how it is. So the first thing we see is that we have around 1% of trap cells, so tomato positive cells. This is the same size of ensemble that we saw when we did single cocaine self-administration. And then when we look at the overlap between the tomato positive cells that we tagged here and the CFOS positive cells that we tagged after the second reinstatement, we see that in the group that had the same reinstatements, we have a stronger, significantly stronger overlap, a larger overlap, than in the group that had only the first reinstatement of cocaine and the second reinstatement of sucrose. And because these are very different ways of tagging the ensembles. With one, we use the genetically modified mice that needs to go through a lot of different steps in able to express tomato and be able to be tagged and visualized. And this one is more like a more direct, okay, we just tag CFOS in immunohistochemistry and that's all we do. It was very important for us to counterbalance the study and start first with the sucrose cue. So if you start with the sucrose cue and open the window with tamoxifen, the first thing you see is that you're going to have a sucrose seeking ensemble, but it's going to be significantly lower than the cocaine one. So what does that mean? And this is lower than the sucrose when it's given as a single self-administration. So what it means is that by exposing the animal to both cocaine and sucrose, the cocaine seeking ensemble remains the same size, but the sucrose seeking one shrinks by half. So this is interesting when we saw, the sucrose, the cue-induced sucrose seeking, that we saw that the amplitude was a little bit lower. It seems that the size of the ensemble is lower as well. But when we looked at the overlap with the CFOS positive cells, we see that again, the group that had two sucrose seeking reinstatements overlaps more than the group that had both the sucrose and the cocaine. And when we look at correlation between the percentage of overlap of ensembles and the behavior, we see that this time we have a negative correlation, meaning the more the cocaine sucrose, the cocaine ensemble and the sucrose ensemble overlap, the less the animal knows what a reward to seek. So it's like the more confused it is. So this kind of tells us that, the more they overlap, the less the animal actually has like a very specific reward that it seeks. So in conclusion, we have shown that we are able to identify a cocaine seeking ensemble during cocaine single reward, and also a sucrose seeking ensemble that I didn't show you, but it's more or less the same size. We're also able to identify an extinction ensemble that is a little bit smaller in the cocaine and a lot smaller for the sucrose. And we're able, with the RNA-scope experiment, if you remember, to identify that most of the neurons recruited in the seeking ensemble were D1 medium spiny neurons, whereas in the extinction ensemble, we had D2's medium spiny neurons, and this would fit the literature really well. And when we looked at the dual cocaine and sucrose administration model, we see that our cocaine-seeking ensemble remains strong at around 1%. The sucrose-seeking ensemble shrinks to half, and the overlap between the cocaine and the sucrose-seeking ensembles is only about 30%, meaning you have mostly very dissociated ensembles for each reward. So with that, I would like to thank everybody, all the founding support. Most of this work was done at the Medical University of South Carolina, where I did my postdoc. And if you're interested in our work, please follow us on Twitter. And of course, please reach out if you have any questions or are interested. I will open the floor to questions.
Perfect. Thank you very much, Ana Clara, for this great talk. I enjoyed a lot. I'm pretty certain that everybody does. So if you want to do any question, please, you can write on the chat, or you can raise your hand and I can habilitate your microphone in order for you to do your question. So if there's any questions.
Okay, so I don't know what I did, but you appear really zoomed in, Alfonso.
Oh, don't worry. If you want, you can stop sharing your screen on the...
Just not sure how to do that.
On the top, you can see next to the microphone.
There you go.
With the window with the next.
Yeah. So it seems like it was blocked. So let me see. I mean, you can ask questions.
Okay. Yes. Yes. If you have any questions, I'm going to ask you a couple of questions. In the meantime, the people are asking the questions. I understand from your conclusions that there's different ensembles in these cocaine and sucrose, these ensembles, right? So this may have some consequences in order to have some intervention on sort of therapy in order to reduce the drug use.
Mm-hmm. Yeah, exactly. Sorry, are you done with the question or?
Yes.
Okay. Yes, exactly. So that's kind of like the proof of principle that we have, that if they are indeed, separated, and we understand the mechanisms that are happening in the cocaine-seeking ensembles, then if we're able to modify those without touching the ones that are the sucrose-seeking ensembles, then we're able to kind of like target the treatment to the cocaine intake, that's the one that is problematic, and not modify the one for sucrose that is a natural reward and that we need to actually, keep eating and remain alive. So it's kind of like interesting to see that this seems to be different networks, and we could eventually target one without altering the other.
Perfect. And another question that is kind of related with that, you mentioned that you are using this cocaine administration. You are trying to use some different drug in order to see if the effect is the same or it could be different in that sense?
Yes, we are, but before doing the other drug that we were interested in is heroin. But before doing that one, the other question that we wanted to answer to first is that, is it the same between two drugs of abuse? So what we did is modify the dual model to have both cocaine exposure and heroin exposure, and see if the fact that they are both drugs of abuse, it also maintains segregated networks, or if it's actually more overlapping. So that's the question that we're trying to answer now.
Oh, perfect. And in that sense, I don't know, maybe this is a naive question, but what happens, for example, for some drug that is not like cocaine or heroin, more like caffeine or something like that? I don't know if it's the same or it works the same.
So I don't think anybody has looked at it. What I'm thinking about a paper that did ethanol, so alcohol versus sucrose, and they show that in another region of the brain, it was the prefrontal cortex, they show that the overlap seems to be a little bit more overlapping, so large. So it seems that it's really it's depending on the type of reward. And so I think that we would find really interesting results if we did caffeine or theine or anything that is not really considered a drug, but it is kind of addictive because you cannot go without it. But that's a very good, interesting question. I think that we're going to do compare drugs of abuse, then compare other drugs with sucrose, and then we might look into more like, I'd say, commonly used drug. Yeah.
Okay, perfect. Manuel has a question, so I'm going to habilitate his microphone. So let me just. Okay, Manuel, you can turn on your microphone and you can ask your question, Manuel.
Thank you very much. Sorry, I have the dental dispositive in my mouth, so I can talk.
Go ahead.
Kind of. Well, my question is, it's there is a difference between the cocaine and some kind of action, like, I don't know, a sport or agree or something that you can make to you addicted, and well, that's the question.
Yeah, okay. Yeah, so you're asking about like the behavioral addictions, right? Like gaming, gambling and sports and things like that. Is that correct? Did I understand correctly?
Yes, exactly.
Yeah. Okay, yeah.
Yeah.
So that's a very good question. We haven't done any work on that, but I think that what could be interesting is some people are putting, you know, like wheels, like running wheels in the where the animals are. And so they show that depending on the breeding, depending on situation, some animals show these characteristics of over-exercising, so kind of like being addictive to exercise, where they're just going to run on the wheel without eating, without drinking, without taking care of pups or things like that, you know. So I think that would be a really interesting question to see, to set that up, like to see, okay, while the animal is exercising, for example, in the wheel, what are the ensembles that are activated, and are those, you know, like larger or maybe stronger than compared to a drug, to a cocaine drug of abuse, right? So that would be a really interesting question. We haven't done any experiments on that, but that's definitely something that is very interesting.
Perfect. Thank you very much, Manuel, for your question. If anybody has another question, I would like to ask another one that arises from that. What happens in, maybe this is not connected, but what happens with depression or anxiety related to this drug abuse or something like this is sucrose, and maybe this with the pandemic and the COVID that increased the depression, and maybe it increased the drug abuse or the use of sucrose. I don't know if there's any connection with this.
Yeah, so you're right. It has been shown in the few studies that have been coming out since the pandemic that, you know, isolation and the fact of having like a very weird and scary context has a high, so we see like more relapse than when we see outside of pandemic. So like people are drinking more, taking more, like it's hard because a lot of people who are trying to recover from drug abuse rely on a very tight network of people, you know, like meetings and things like that. And those, I mean, mostly don't happen. Like they're trying to move to online, but it's like a very slow change. And so, yeah, so we do see an increase in relapse. And I think that anxiety and depression definitely are going to impact those ensembles. And probably like either potentiate them when they are more activated. They're going to be more activated than usual. But we haven't really done so far any experiments where we put, for example, the animal in a depression-like or anxiety-like context, and we see how it impacts the ensembles. We do see, like a lot of people have seen that if you make the animals more anxious or more depressed, then they're gonna take more drugs, they're gonna seek stronger, the restatement is gonna be heavier and more robust. We do know that. So what might be happening is that when you put the animals in those contexts, then you kind of like super activate the ensembles and then make the craving for those drugs even more appealing and harder to avoid, you know.
Okay, perfect. Thank you very much. I don't know if anybody has another question. Well, Manuel say thank you very much in the chat. And I would like to ask you if there's another question, but I'm going to wait a little bit. But I want to ask you this question I always ask for our speakers. As I told you at the beginning, mostly of our students are female students. So we are trying to motivate them in order to seek these opportunities, talk in front of the people, to visualize the research that the female scientists are doing. So I would like to ask you for a final message for them in order to get motivated for this career, the science, and that you could talk to us about a little bit about the background that you need to have in order to do some research in the area that you are doing. So I would like to ask you for this message for them.
Yeah, so that's a very, it's a very interesting question and good question. I think that it's definitely harder for women in science or for any type of minority, you know, that is not basically just like old white men. And it's because of society, like it's that the way that society has been developed. But I think that we're pushing through, like we're coming strong. And it is harder. It's going to be harder for us. You know, I'm an immigrant in this country, so it has always hasn't been easy, you know, with the accent and not understanding everything and being a woman. But I think that my family and my mother in particular, who was also a researcher, has just taught me that, you know, you're as smart as anybody else. You have to keep going. You may have to work a little bit harder, but if that's what you really want to do, you can do it. And so we're just, you know, taking the high and the lows and just taking the time to absorb everything and then keep going. And like you said, Alfonso, it's very important to take advantage of every possibility. So when you reached out, I'm like, yes, let's do it. Like, do I want to give a talk? Do I want to prepare? Do I want to do all this? No, like I'd rather stay home and do other things. But this is important because this is going to give you visibility. This is going to increase your network. And this is going to also allow other people to see that, yes, you can be a woman in science. You can do interesting research and just do like, you know, other big majority of people, like white people, I guess, white male, do, you know, like you can do the same. It's just going to be a little bit harder, but that's what you really want to do. Keep pushing through, and the more people do that, the a little bit easier it's going to get, I guess. So just like keep going, and if you want to talk more about my personal experience, or if you have questions or stuff you want to share, please reach out through email. I'm happy to, you know, have Zoom chats or email chats and kind of discuss. I think that the network is probably very important. So go through what happens and just cheer for each other, and being there for each other is very important.